So in this video we will discuss about the complementary proteins. Complementary proteins. What are basically complementary proteins? Basically these are the small proteins. These are the small proteins that help the monocyte to kill pathogen. That help monocyte to kill the pathogen. Basically it is produced by the liver. Complementary protein is produced by liver. If you talk about the range of complementary protein, it produced 20 to 30. Average, if you talk about that, is 20 to 30. So it is present in inactive form. Basically it is present in inactive form in the form of plasma fluid. Present in inactive form in plasma fluid. So if we talk about this, when what we can say function of the complementary proteins. So when any pathogen will enter, it will get active. It is inactive as I written here, right? So when any pathogen will enter, it will get active. It will get active. Basically if it are complementary proteins, it's somehow like chain. It is like this. It is chain structure. Basically if this will get active, so this will get active and this will do active to this, and this will get active to this, and this will do active to this, and this is a whole chain like this. If one will get active, whole will get active. One will active to each other, another will active to other, another will active to other. So that is basically the chain. So that will be the active chain. So if suppose that we are having here cell, right? So when we can say when pathogen will enter in, it will get active. So this protein, complementary protein, will do holes in plasma membrane. Suppose this is a plasma membrane, so it will do holes in plasma membrane. When it will do holes in plasma membrane, so this will fill water and salt in this cell. In this cell, it will fill water and salt in this cell, so that we can say osmotic potential will be imbalanced. So osmotic potential will get, we can say, imbalance. When it will get imbalanced, so cell will burst. When the, we can say, cell potential or cell fluid will get imbalanced, so the cell will burst. So that was the, we can say, function of the complementary protein. That will, we can say, first it will be in the inactive form in the plasma fluid. So when any pathogen will enter, it will get active. If one will get active, it will do active to we can say whole chain. So this was a function of the complementary protein. If I told the complementary protein is a non-specific, it is a non-specific. So that was all about complementary protein.